All right. Hello, everybody. Hello. Hi. It is 10 o'clock, so I wanted to get things started here. My name's Tatiana. I will be introducing just what's going on today, and we'll get started with our first oral session in a few minutes. For those of you that are joining us today, hello and welcome. We've got food and refreshments in the back, so please help yourselves to that while anything is going on today. We will have a few breaks throughout the day, as well as lunch, so those are all things to look forward to, with events just happening between 10 and 4 today. So with that, I also wanted to welcome everybody that is joining us online today. We have presence today on Zoom and YouTube, so anybody that is joining us can also ask questions on there to our speakers. We'll get into those details in a little bit. Make sure I'm okay. So if you didn't get a chance to meet any of the folks that are organizing today, we are organizing this event, the ExplOrigins Colloquium of 2023, on behalf of the Georgia Tech Astrobiology Program and the ExplOrigins Early Career Group. We are a group of early career scientists that are all interested in the search for life, explorations and origins, so exploring the universe, studying the origins of life, and we meet on a regular basis. Our meetings this semester, if you would like to join us, if you are someone here that is at Georgia Tech that would like to attend our meetings, are happening every other Friday at 1 p.m. So we have both in-person and virtual attendance available for both of those. And if you'd like to join our Slack, where we do a lot of communicating for where ExplOrigins events are happening, you may scan this QR code, and it's also accessible on the Georgia Tech Astrobiology website. We wanted to put a plug here for the Georgia Tech Astrobiology Fellowship. For those of you that are graduate students at Georgia Tech doing a master's or a PhD, this is an opportunity that is designed for you and available to you to apply to. So this event right here was co-organized by ExplOrigins and this year's Georgia Tech Astrobiology Fellows. And the application for next year's fellows, or I guess the 2023-2024 fellows, is going to be opening this month, and it is due late April of 2023. This is an opportunity to represent the Georgia Tech Astrobiology program. It is a fellowship, so you get a $4,000 fellowship through it, and you help organize events on behalf of the astrobiology program. So please apply to that. We have it linked on the website as well, and we are looking forward to getting to know who next year's fellows will be. This is not updated, but if you want to view our program, for those of you that are in person, we have a few flyers that are available to scan the QR code. We're mostly doing things virtually, but we also have some paper programs in the back as well. And for those of you who are joining us online, we will paste a link in the chat to the virtual program as well. We are really looking forward to our plenary speakers, Dr. Emmy Williams and Dr. Heather Abbott-Lyon, who are joining us in person today, and they will be giving talks at 1:30 p.m. Eastern Time and 2:20 p.m. Eastern Time. So we're especially looking forward to that as well, along with the 3 oral sessions that will be hosted by early career researchers here at Georgia Tech and some online presence as well. So this is the timing for our session 1, which we will get started with momentarily. Did anyone else have anything they wanted to add? Yes. Yes, that's correct. Yeah. Oh, thank you. Thank you for clarifying. Yes. Yes. Ooh. Unless anyone unless anyone has a hashtag they want to propose right now. I like that. Perfect. Yeah, hashtag ExplOrigins23 if anybody missed that online. Thank you. And the program is available in the Zoom chat for those of you online. Okay, so I would like to just give an overview of session 1. We have 3 student speakers, which are Sharissa, Emmy, and myself, Tatiana. So I'm going to get the slides ready, and Sharissa will be our first speaker. I ask that folks save their questions for the end. So if you're in person, please raise your hand at the end and we will bring a microphone to you. If you are online, either on YouTube or on Zoom, please enter your questions in the chat and we will have somebody relay them to us in the room right here. And Sharissa, if you see questions online, just wait till the end and we will use the microphone to ask them to you. There you go. Oh, to use the microphone too?
Okay, awesome. Well, good morning, everyone, and welcome. I'm excited to be your first speaker today on this lovely Friday morning. For those joining online and who don't know me, my name is Sharissa Thompson. I am a PhD second-year student here at Georgia Tech, working with Dr. Rivera Hernandez. And so my work focuses on Mars laboratory analog sediment flows, investigating the effects of pressure and water-rock ratio. And so with that, let us begin. So due to geological features observed on Mars by both orbital and rover-based data, we see evidence for liquid water persisting throughout Mars's history. As you can see here, I have evidence of gullies on Mars that need water in order to propagate, as well as alluvial fans that have water create them. And then this is a Earth-based comparison of a mud volcano that involves a little bit of water with a lot of sediments within the flows. And so on a geological timescale, conditions such as pressure, temperature, and water availability have varied on Mars. And currently, diurnal temperature shifts as well as changes in elevation causes a change in temperature and pressure as well. So commonly, planetary geologists use terrestrial analog experiments or comparative Earth studies to better understand the features that we are observing on Mars. But what these studies commonly lack is the conditions that are indicative of current and past Martian climate. A few studies have taken this fact into consideration, including one conducted by our collaborator and co-author, Dr. Broge. So in previous studies, we see that with the decrease in pressure, there is this impact on the deposit morphology of flows. And for Dr. Broge, pictured here, his studies use bentonite clay mixed with magnesium sulfate salts under 5 millibar pressures inside of a pressure chamber to simulate the pressures that are currently on Mars today. And these sediment flows ran, and a pressure caused the boiling effect to occur. And this boiling process, also known as vaporization, resulted in bubbling as well as levitation behaviors. And notably, these behaviors cause a longer run-out deposit morphology. So what this means is that the importance of deposit morphology for the features we are observing on Mars today is important to note that the deposit properties is used to better understand the surface processes and hydrological activity that we have observed on Mars that could have happened in the past. Therefore, it is necessary to constrain under which specific conditions such behaviors occur, and applying such constraints inspired our work that we did for these experiments and trying to understand the impacts of early Martian conditions on these flows. And so in turn, we came up with the following questions. The first question asks, at what pressure temperature conditions do we see these bubbling and levitation behaviors occurring? Oh, sorry, I flipped those. But at low water rock ratios, what do we see happening with the bubbling and levitation? And then at what pressures do we no longer see this boiling process and the bubbling and levitation behaviors? And so the importance of this lies in the fact that compared to Dr. Broge's study, we will be using sediments and water availability that is more commonly found on Mars, as well as our pressures will range higher due to the difference in pressures in the past compared to current Martian pressures. And so in order to do that, we varied a couple of parameters. And the first parameter that we varied was temperature. And in order to do that, we changed the temperature of the sand bed that these flows were propagating along. And for the warm beds, we set them at 293 kelvins in order to simulate a sort of Earth temperature. And for a Mars-like temperature, we froze a sand bed inside of a deep freezer in order to get 270 Kelvin temperatures. We also varied the water-rock ratio. We used lower water-rock ratios as compared to Dr. Broge's. So we had 30-70 water-rock ratio and a 40-60. And this is relating to the water availability on Mars, indicating that there was less water possibly available in the past. And this is trying to simulate what kind of water would impact the flows and how much of that water would be needed in order for it to propagate and what that would look like. We also vary the pressures, as you see here, from 7 millibars to the current Martian pressures today up to 1013 millibars, which relates to our pressures here on Earth. And as you can see, current Martian pressures are about 1/100th or 1/1000th of what we have here on Earth, just to put that in your mindset. And over here pictured on your right, I have our experimental setup inside the chamber. So we used the UK Open University pressure chamber to create our experiments and run them in. And we had 3 DSLR cameras set up inside of the chamber in order to take video captures of the flows as they were propagating along the sand bed. And as mentioned before, we used basalt sediment in order to kind of relate to material that we more commonly find on Mars today. And we used this is our baseline experiment. So we wanted to remove any extra ions or other factors that may interact with the water and cause the flow to be impacted. So we just used regular basalt Exolith lab simulant in order to try to have this base level of a flow experiment. And so the flow bed was at a 10-degree tilt along a unconsolidated fine grained sand bed. And that is something to note because the sand is unconsolidated, and you will see that had an impact on our experiment in my next slide. So here is a video of one of the low sediment rock ratio or low water rock ratio, low 5 millibar pressure, but warm sand bed experiment running. And hopefully, yes. Okay, so for this video, I would like you to note that the aforementioned vaporization process we see, as well as this bubbling and levitation occurring, you can see that the sand grains in this video sort of saltated or were lifted up from their original deposition and redeposited on top of the sediment flow that we have running, the basalt flow. And you can kind of note that from the way it's covering up the end of the flow, as well as, I don't know if anyone paid special attention to it, I could probably play it again, but there was that sort of levitation occurring where it's having the vaporization bubbles between the sand bed and the sediment flow cause air to get trapped between them and allow the flow to propagate further than we would have expected originally. And so for the next couple of slides, I will show you our experimental results, and those will help answer the main questions that I originally proposed. So for our first result, you are seeing that at 5 millibar pressures under warmer bed flows, vaporization causes a magnified bubbling and levitation. As seen in the previous video and along the top of these images, you can see that saltating of the sand grains covering up the flow is equitable to the mixing and bubbling and levitation of behavior that I mentioned before. But this was not seen as much in colder sand beds. So this is indicating that in colder climates and under colder temperatures on Mars, we wouldn't see this happening. But if it was a warmer climate, as we see or possibly think there may have been in Mars's past, this means that this behavior could have been occurring and affecting the surface processes that we're observing. And so the next result that we noted was that, again, at 5 millibar pressures, we see a longer run out for the high water rock flows. And this is more predominantly seen in the warm beds. As previously stated, the warm beds have this behavior of levitation occurring, causing the run out deposit morphologies to be longer compared to these more stunted frozen flows. And so this result, our third result, showed that even under 5 millibar pressures, the lower water rock ratios have a thicker deposit, and this was magnified in colder beds. So this top image, or the top 2 images, are showing the warmer beds, but the saltation and the mixing of the sand bed with our basalt flows made it more difficult to define the actual thickness and whether or not it was the basalt only creating that thickness or whether it was the mixture of the sand bed with the basalt creating a thicker flow. So it's easier to define that variation in thickness for a lower water rock ratio in the colder beds than it is the warmer. And so the next experiments will go over changes in pressures and how that impacted our results. So for this one, and very importantly, bubbling and levitation only occurs for pressures less than or equal to 14 millibars. This is important for constraining past pressures on Mars and what they were at what specific times by notably observing the surface processes or the surface features that we see from orbital and rover data, and being able to say that one is longer or one has this mixing, and that probably happened under lower pressures than those that don't have this mixing behavior occurring. And so for the next slide, it goes over this sort of bimodal distribution. So as you see on the bottom, it has the 5 millibar pressure and with the mixing and bubbling occurring. And at the top, it has the 10-13 millibar of earth pressures. And you can kind of see that the lowest is long as well as the highest, but in the middle, we sort of get this dip of it gets shorter again. And I think that is due to the 14 millibar pressures not having that levitation and bubbling occurring as much. So it is creating a more stunted flow, even though the pressure is increasing overall. So we have this bimodal distribution of the length-width ratio that we noted in our experiments. And lastly, the lower pressures cause more lobes to form. Interestingly enough, under Earth pressures, there was a singular lobe, typically elongated, propagating out in front of more stunted leftover sediment. Whereas with the lower pressures, the 5 millibar and 14 millibar, you see all of these lobes forming underneath these lower pressures. And that was interesting to note because, again, this is indicative of if we see features on Mars with an abundant amount of lobes, this means it probably formed under lower pressures than what we would expect at 20 millibars. And so to conclude on my take-home points, in answer of the first question, our preliminary results are supported by Broge et al. And our observations are similar to his regardless of the water-rock ratio and vaporization that causes the levitating bubbles. So even though we varied our sediment as well as we used more water in our experiments, we found that our results were similar to the earlier experiments run by Dr. Broge. And to answer the second question, bubbling and levitation was only observed at 14 millibars or less for high or low water rock ratios. And so this is just running through all of the points I touched upon earlier, that resulting deposit morphology was impacted by bimodal distribution of the length times width ratio with increasing pressure. The deposit thickness was impacted with increasing lower water rock ratios, as well as lobe quantity increase with decreasing pressures. And so that's the end of my talk, and thank you for listening.
Yeah, so I actually do have a question, which I'm going to just go ahead and ask. Do you have any sense for, as this bubbling and levitation process is going on, how high are the grains levitated above the surface? So how far can they reach beyond sort of that narrow area where the water track's at?
That is a good question. So we didn't measure this quantitatively, but we do have a side angle video of the grain saltating as the flow is propagating. So if we wanted to, we could use the scales in our images to try to constrain how high that distance is. But roughly, I would say it's a good couple of centimeters high that these grains are saltating into the air.
So then they're going to reach like a couple centimeters or a couple of centimeters away as well.
Yes.
Cool.
Yeah.
Very cool.
Great talk. I guess I'm wondering what sort of, I guess, grain size and for the water cohesion adhesion limitations might you have doing the experiment at this scale versus doing it, or comparing it to meter or kilometer scale? Because I would imagine some sort of the bubbling might just be different because of the cohesion adhesion.
Yes, that is a really good question. So I didn't mention this, but we used deionized water. So there wasn't much cohesion of the grains with the water. There was no salt to bind them together. And we didn't use clay like Broge did. So there was no swelling combining the mix to keep together. So what ended up happening is we used a blender inside of the pressure chamber to mix the sediment flows as the pressure was decreasing to make sure that it was homogeneous in order to keep the flows like that together. And as far as scale and scaling this up to kilometer size, we would expect that with a larger size scale that we wouldn't see as much of this mixing going on with the propagating flow with the underlying substrate sand bed. And we do have interest in running further experiments by changing our substrate and using either sandpaper or some other form that is not unconsolidated, so we don't have that impact on our experiments.
Thanks. Other questions? It's where I can see people. Yeah.
So you picked 270 and 293 as your temperatures and your pressure ranges. Do you have some idea about time periods where those may have been relevant surface temperatures on Mars? Or do you have some idea about what the pressure, like, is there a particular time that you're sort of comparing your experiments to? Because there's obviously not free running water on Mars's surface now, right? So at some time in the past, is there other data that you could use to sort of constrain what those pressures and temperatures should be?
We currently do not have specific data to constrain it. This is kind of working at constraining that. We're just basing it off of the past 3 billion years on Mars, and that's why we had such a wide range of pressures to try to see under which pressure we do see these flows, and then try to constrain the pressure and time to that.
Thanks.
I think we're a little ahead of schedule. Got one? Yeah.
Ooh, yes.
Very dumb question. Okay. I'm just kind of curious, like, as you see like your differences in your pressure, did anyone run the experiments at Earth ambient pressure just to kind of see like how similar those flows would be to what you might expect to see on Earth, and just use that as sort of a benchmark for how different things are in your experiments?
Mm-hmm. Yes. Yeah. So the top pressure that you saw on the screen, I can go back to that 1013 pressure. It's kind of covered by the Zoom thing. Is the Earth ambient pressures that we are simulating to compare to.
Yep. I totally missed that. Sorry.
That is okay. Yes. Darn Zoom.
Okay, cool. Let's thank our speaker again. So I'm going to be presenting the next couple of talks here. And I do want to point out that this is kind of a cool first session because all three of our speakers are actually our current astrobiology fellows as well. I think the fourth astrobiology fellow is off in the ocean right now. And so she's not able to be here today, but it is kind of a cool, the fellow session in this first one. So our next talk is going to be by Emmy Hughes. Emmy will be telling us today about in situ analysis of sodium sulfates as paleoclimate indicators for Mars. So Emmy, thank you. Take it away.
Thanks very much, Micah. Hello, everyone. I'm Emmy Hughes, as introduced. I'm a second-year PhD student here at Georgia Tech, working with Dr. James Ray and Dr. Francis Rivera Hernandez. And today I'll be talking about, as Micah said, modeling and in situ analysis of sodium sulfates as paleoclimate indicators for Mars. So why are we interested in something that will tell us about the paleoclimate of Mars? Well, Mars may have been warm and wet, or cold and icy, or cold and dry in its early history. And it's one of the fundamental questions about the history of this planet. So what are the tools that we can potentially use to constrain these conditions? Well, there are a number of things. There is modeling, there's using in situ data, and there's looking at salts and looking at different kinds of minerals. So we know that lakes were present on early Mars. We know this because we have geomorphic evidence, and we also have mineralogical evidence for such lakes existing, but we don't know what they were like in terms of their temperature and their environment. And it's possible that these lakes were stabilized via an ice cover or something that kept ions or dissolved gases under the surface of the ice, such that the brine may have been stable for longer periods of time rather than just intermittently. And we can potentially use visible to near infrared spectroscopy to observe evidence of such lakes. So this is data from my advisor's paper using visible to near infrared data, looking at different kinds of minerals, including sulfates at the surface of Mars, and potentially using those as paleoclimate indicators. So I'm focusing specifically on temperature here. So I'm looking at salts as possible temperature indicators for early Mars. And so there's a couple of different things that we can look at. We can look at first evaporative versus freezing processes of cryoconcentrating a brine and producing precipitates. And that gives you different sets of minerals as a result of those two processes. So if we look here on the left or on the right here, where's the laser pointer? Is it this one? Oh, I found it. Okay, great. So under evaporation of a baseline seawater brine, the first thing that you get out of it is aragonite and then gypsum. And by the end, you get these bittern salts that are potassium and magnesium rich. If you freeze that solution, however, the first salt that you get is mirabolite, which will be the subject of this talk in part, which is a sodium sulfate decahydrate. And then you move into these really highly hydrated magnesium and potassium chlorides. So under this system, you essentially see things with these higher hydration states, and you get mirabolite precipitating out earlier. And then over here, I also have a phase diagram of the sodium sulfate magnesium sulfate system where we see at 25 degrees C, there's bloodite, which is a mixed magnesium sodium sulfate that's stable, whereas under 0 degrees C, we do not see the stability of bloodite. So it's just mirabolite and epsomite, which are sodium and magnesium sulfate end members. And then over here on the far left, I'm showing a picture from a place that I will be talking about for the rest of this talk, the Basque Lakes, which under this very cold temperature, I'm not sure if you can see it on the monitor, there is meridianite forming, which is named for Meridiani Planum, where the Opportunity rover explored, where we saw evidence for these magnesium sulfates with 11 hydration states. So very, very highly hydrated salts that only form under very particular cold conditions. So the fundamental questions guiding this work and this talk are how the environment and formation mechanism will influence assemblages of minerals forming from a brine, and how we can rely on the differences in mineralogy to reconstruct that formation environment. So we can rely on things like known parameters about places that we have a lot of access to data for. So like Earth, we have a lot of potential for constraining a lot of the variables, including the temperature, the relative humidity, potentially the geologic context. And then we can get data that is Mars relevant in those environments and be able to then back kind of constrain what they were like on Mars. So that's looking at things like the crystal habits, spectral signatures in the visible to near infrared, and relationally the geochemistry. So that brings us to the Caribou Plateau, which will be the subject of this talk, and also the place from which I got off a flight at 5 a.m. this morning. So forgive me if I'm a little bit, if there's any typos or anything, just don't tell me. So over here, we're looking at Last Chance Lake. This is my collaborator, Jacob Buffa, walking out onto the lake in the morning. This may look like ice to you, but it's actually all salt. So we've got these kind of edge salt deposits, and then these brine pools with enigmatic origins forming in the center of this lake. So it's unclear exactly what process leads to these kind of spotted formations, but we see a really, really hypersaline system here with a lot of precipitation in the summertime and then freezing over in the wintertime, where you get these cryoconcentration processes and different sets of salts. So this lake is Last Chance Lake. The other lakes that I'll be talking about are the Basque Lakes. So these have two very different compositions. Last Chance Lake, I'll talk about later, is a carbonate-enriched lake, very alkaline, whereas the Basque Lakes are very enriched in magnesium and sulfate. Magnesium sulfate, of course, being a highly relevant salt to early Mars and potentially the Hesperian era. And I think Sharissa mentioned it also in her talk as well. So these lakes have a ton, as you can see in this concentration, just a ton of magnesium and sulfate, which makes them very interesting for that system, as well as high sodium, which is also relevant. So I'll be talking mostly about Basque Lake 2. This is this lake here. You can see that there are spots forming already in this aerial image. And then on the surface, you have varying degrees of water over the course of the year. So in this picture, which is taken from 2019, we have quite a bit of water forming in the center of the pools. When I visited both in October and in, this is October, both in October and in February, it was much more concentrated. So here we're looking at a salty surface, and here this is actually ice, and you can see it's the same angle there. So the original understanding of this lake from 1918, so over 100 years ago, when they were being considered for mining, which didn't work out very well. There's an epsomite layer, and then there are these sort of black muds that form the ridges. So here's the epsomite, that's magnesium sulfate, that should ostensibly be under these brine pools. And then these sort of raised features are these black muds that they say. And then there's a layer of gypsum supposedly, and then metamorphic rock as the basement rock. So the subject of this talk will involve a little bit of modeling using freeze chem. So that's a geochemical low temperature aqueous model where I'm looking at what precipitates we should see under the summertime and under the wintertime. And then I'll show you some spectra from each time of the year and see kind of how it compares to the freeze chem model. So these values in the dashed lines are the winter salts that should be forming, and then these in the solid lines are what should be forming in the summer. So you see from freeze chem that we should have a significant amount of meridianite in the wintertime, and epsomite is the stable sodium sulfate or magnesium sulfate in the summertime. And then if we look at our final or very close to full evaporation weight percent pie charts here, we see a lot of epsomite in the summertime and then a lot of meridianite and mirabolite and very small amounts of other salts that I didn't include on this, but you can see them here, so including gypsum. So these spectra are from the summertime. So we're looking at these kind of fluffy efflorescing crusts that form on the edges of the lakes here. And we can see quite clearly from the spectra that it's epsomite, that it's a magnesium sulfate salt forming. We've got a lot of the absorptions accounted for here, including the sulfur hydrogen specific bonding is showing up in the spectra. So looks like we're absolutely confirming in the model that there's a lot of epsomite. And also in the model, I include a couple of other salts that we should be able to see that we're not seeing as easily. So epsomite is quite dominant or quite overwhelming in the spectra here. If we move to the winter time, it's much more difficult in the winter to get data from these because a lot of them are forming under the ice. And so when you pull them out, most of the signatures that we see are hydration, and they're very, very, very strong in the visible to near infrared. So it's hard to make out salt-specific features. But we did find this log here that had these kind of crusts also forming on the log. And this is a place where known meridianite may have formed on these lakes previously. And what we see is something more like maybe mirabolite. And so that's from this signature here, the 1.77 feature is associated with mirabolite, not with epsomite. So it looks like we might be forming sodium sulfates there on the crust. Now we move to Last Chance Lake. So Last Chance Lake, as I noted earlier, is very enriched in calcium carbonate or in carbonate ions and very high amounts of sodium, not really calcium, sorry. So they're mostly a sodium carbonate and then a little bit of a sulfur and chlorine enriched lake as well. And so these have a slightly different basement rock. There's basalt and there's till that's just on top of that. And then you've got the lake and a couple of mud flats. And so this is an older paper as well. Much of the mineralogy from these areas were done in the 20th century, not so much more recently. And this is what the lake looks like here. So you can see it's still got those spots. So the modeling for Last Chance Lake indicates, again, a different set of minerals under each set of conditions. So we see high nacolite and dolomite, calcite, and mirabolite in the wintertime. And in the summertime, we see a lot of trona. So trona is sort of a mixed bicarbonate, carbonate, sodium salt. Nacolite, I think, is just sodium bicarbonate or sodium carbonate, 10-hydrate maybe. And then, yeah, we see things like magnesite and aphthiolite and other salts that should be forming in the summertime. So then looking at the spectra again, there were two really distinct mineral habits at this lake. Forming in the center of the brine pools were what we termed the almonds. They're these sort of bladed salts that would form in clusters. And then on the ridges, you would get these botryoidal sort of fluffy ridge salts. So these were quite hard to characterize again spectrally. We're looking at a couple of spectra from those there in the blue and then in the gray. So they might be almost a wet trona. So we're seeing kind of absorptions here and here. They're kind of pulling down, possible water hydration, very strong there. But the salts that are forming on the fluffy crusts, again, look a little bit more like mirabolite, again, with that 1.77 feature. So it's possible that we've got a slight difference in salt assemblage between the ones that are forming on the edge and the ones that are forming in the pool, but it's hard to tell with the hydration. So we'll have to look into X-ray diffraction and other a little more mineralogically rigorous methods here at the home front. Finally, we have this winter core that I just pulled out a couple of days ago that I'm showing a little bit of spectra from. So it's quite, it's got a lot of mud in there and quite a bit of algae, sulfate reducing algae as well. So this is what those spectra look like. And again, it looks a little bit closer to mirabolite than the other things that are predicted to form. So one critical thing here is the only salts that we sort of definitively see in these sodium carbonate rich lakes looks like a sodium sulfate. It's hard to identify these sodium carbonates in the spectra. So just to do one quick spectral comparison here, looking at Basque Lake 2, we've got, again, these fluffy salts here that are forming on the edges in the summer. And then we've got these that are forming on this kind of piece of wood in the wintertime. And we're seeing totally different spectral features here, where you've got these kind of strong 1.7 in the wintertime salts, and then this 1.63 that's closer to epsomite in the summer crust there. So could be some sort of conversion, could be something that's forming differently in different areas of the brine pools. Many questions that have yet to be answered. So just as a quick note, there's also a stable anhydrous sodium sulfate phase that can form in really low relative humidities and higher temperatures. All of the relative humidities and temperatures for this region are consistent with epsomite, at least in the wintertime, or mirabolite, at least in the wintertime. So that's what we should be expecting to see, and that is what we're seeing, which is great. So mirabolite was likely detected for both the cold and warm conditions at Last Chance Lake. There were no obvious spectral presence of sodium carbonates in the spectra. Sodium carbonates have also not been spectrally identified on Mars. And it's possible that this is not due to their absence, but due to some other spectral mixing process. They also don't have some of the characteristic 2.3 and 2.5, mainly the 2.3 features that you expect to be associated with carbonates. So epsomite was likely detected under warm conditions for Basque Lake 2. Mirabolite seems to have been detected under the colder conditions, but meridianite I could not find a spectrum for when I was looking in the last couple of days. So if anyone knows where I can find one, that would be great. But in the meantime, I'm going to keep questing after that. It seems like the presence of ice extensively obscures minerals. This is understandable because you've got these strong, broad hydration features that form with ice, but it'll be interesting to keep looking at those salt-specific hydration features and see how they mix spectrally. And then finally, as I noted earlier, mirabolite detections may not preclude the presence of carbonates, and it's also possible that there's these seasonal changes in the crust composition of these lakes. So thank you, and I will take any questions.
Thank you. Yeah, hold on. Got a couple questions here. Tony.
Hey, interesting stuff. I was wondering, are there in these brines that form this stuff that you are getting all these spectra from, are there microbes in these brines, and could their presence affect what precipitates out?
Yeah, so there are sulfate reducing microbes that are present in sort of a layer just underneath the top layers of salt. And so they seem to be really happy at the interface between the muds and the salts. So it seems like, taking advantage of the water, taking advantage of the chemical energy. My guess would be if they are influencing it, it would be in the production of salt. I mean, there's the HS gas that they're doing, but I don't think that they're sufficiently altering the mineralogy such that, there's a low abundance of sulfate.
So we're not talking the pink salt flats that are full of dead bacteria.
Yeah, we're not talking that. I think if it's going to show up in the visible to near infrared, it's going to be that spike in the green, but otherwise I haven't seen anything that's very clearly like that's a spectral life signature other than that.
Okay.
But I'll keep looking.
Cool. Got one over here. Excuse me.
Awesome talk. So I guess I don't know a ton about these spectral analyses, but so you did these in situ?
Yes,
Okay,
these are in
cool.
situ. Yeah.
So I guess I don't know if this makes any sense or not, but if you were to take the minerals that you got there in the winter and then bring them to an 80-degree environment, like I imagine you're now analyzing the ones in the summer in, could just warming them up, on the day of, not during their formation processes or anything, could that change the spectral signature at all?
It's a really good question. I think the answer is yes, potentially. If they're really unstable, what they'll do is essentially melt. They'll just lose their water and it'll kind of borderline sort of deliquesce come off of them. The real thing that would impact that is the kinetics. So if you've got something that's really crystalline, it takes a little bit of a longer time for it to alter as it changes in the environment. If you've got an amorphous salt that's forming, and I took it from here, I kept it cold, I brought it back in the summertime, I let it sit there, it's possible that on very short timescales it would alter. Some of these, salts will form and then will dissolve over the course of a day. Sometimes here, you'll get these sort of surface crusts that then dissolve again. So it can be very quick. So it's a good experiment. We'll see if that ends up happening.
How big was your field spectrometer?
How big was this? It was pretty small. It's like this big. Yeah. So I was wearing it in my backpack with I wish I had a picture. I kind of went around like shooting Yeah.
Other questions?
Really excellent talk, Emmy. Thanks. So I liked the microbiology question is sort of like a lead in. So I've heard about Basque Lakes in the sense of the microbiology work that's been done there. But do you know of anyone who has looked at the organics preserved within the salts and the muds?
No.
Okay.
Don't.
Did you know that I look at organics in salts and muds? So
Could be really good.
yeah. Because I mean, because of the relationship, of course, with like magnesium sulfates that we see on in Gale Crater and everything. Like, there's been a lot of curiosity about how detectable the organics would be in that kind of mineralogy using the techniques that we have on board, like Curiosity, for instance. So if there are samples that are sitting around, that would be something to maybe talk about during the break.
We should totally talk about it. There are samples, and they do have sort of occasionally trapped kind of biological material in them that you can see being preserved in the salt crystalline structure.
That's
so
really
it'd be
exciting.
Really cool to talk about it more with you. Yes, I haven't shown, so I don't yet know how to interpret XRF spectra just by looking at it, but I have a lot of XRF spectra to go through that could be fun to analyze. Yeah.
Fabulous.
Yes.
Continued analysis. Yeah, we'll have to get an update next year.
Yeah.
Other questions? Yeah.
So you mentioned when you were looking at your spectra that you might potentially do X-ray diffraction on some of your samples to try to identify them more definitively. That can be kind of challenging if they're amorphous and also if they contain lots of waters of hydration.
Yes.
Have you thought about how you'll tackle some of those challenges?
Yeah, so I think I totally agree with you on the amorphous thing, and that's one of my that's totally a caveat with the XRD. So I think the way to get around that to some extent is to do the in situ visible to near infrared, obtain the libs and the XRF data, and then sort of try to get at a kind of a cohesive comprehension of what they are. Some of these are quite crystalline, though. You know, they have like very clear and somewhat, at least, you know, relatively stable structures. So I'm hopeful that we could get those to work. I didn't know that really high hydration states could also influence.
You can try drying them mildly. So not really desorbing the waters of hydration, but just sort of heating them. Because sometimes if they have waters of hydration, then they also will hold water on the surface, which is different. It's not like really trapped in the crystal. So you could try doing like a mild heating. And the other thing is for X-ray diffraction, you don't have to have 100% crystallinity, right? So as long as it's got some crystallinity, you'll probably see.
Excellent. These definitely have some of them anyway. Yeah. Okay. Thank you for the advice.
Messy. So I want to ask one more question real quick, which is it's hopefully a little bit leading because I was curious, do you have any sort of planned laboratory work to sort of test these mineralization processes where you can sort of, you know, get at this question that Tony asked is what is the influence of the biology, sort of what are the kinetics? Like, do you have any planned lab work that you're going to be doing?
Yeah, we do. Yeah. Well, it depends on if it gets funded, I think. But yes, there are planned experimental and freezing laboratory analyses that will study exactly what you're asking, the kind of baseline, you know, down to brass tacks sort of changes that we should expect. And then we'll have a nice comparative data set.
Cool.
Yeah. And under Mars conditions. Yes. Yes.
Okay, let's give Emmy another hand. Thank you. If you haven't noticed yet, our first session is sort of our Mars session here. And so finishing us off is going to be Tatiana talking about some research that she's been doing with the Gale crater. So I won't read your title this time. I'll let you do it. Take it away.
Thank you. One moment here. All right. Yes, so this is my talk on using rounding to reconstruct the depositional and transport histories of conglomerates in Gale crater on Mars. I'm Tatiana. I am a second-year PhD student working with Dr. Rivera Hernandez here at Georgia Tech, and this is the work that I've been doing with rover data. Okay, there we go. When the Curiosity rover first landed in Gale crater in 2012 on Mars, only a few weeks within the start of its mission, it detected conglomerates. It took images of these sedimentary rocks with gravel-sized clasts of diameters of 2 millimeters or larger. This was really exciting because it provided the first in situ evidence for flowing water on the surface of Mars. Mars today does not have a climate that is able to support temperature and pressure conditions to allow liquid water to flow at the surface. So these conglomerates provided really exciting evidence for flowing water in the past. We're talking billions of years ago in the past. This meant that we were able to take observations of the rounding of the clasts, and that was one of the key indicators that these were deposited by rivers. Fluvial transport, so deposition and transport by rivers, is abrasive, so it allows clasts to be rounded when they're transported in river channels, and that's how this initial interpretation was made. This paired really nicely with observations that were made of Gale crater by just decades of orbital data as well. We see this feature in Gale crater known as the Peace Vallis fan, which is highlighted there in yellow. And this is intentionally where the Curiosity rover was landed, at the base of the Peace Vallis alluvial fan. This is an area of sediment deposition that was already thought to be deposited by water, and the conglomerates help to confirm that. But you can also see the Peace Vallis channel that leads to the rim of Gale crater. So we have a sediment source at the crater rim, transport down the Peace Vallis channel, and deposition on the fan. And this is what the conglomerates are able to show us. Based on the sizes of the clasts in the conglomerates themselves, the original research in 2013 was able to construct a paleo velocity ranging between paleoflow velocity ranging between 0.2 and 0.75 meters per second, giving insight into the paleohydrology of Gale crater, which gives us insight into the conditions and the nature of the water that was in Gale crater in the first place when liquid water was stable at the surface. So this initial work that was done in 2013, looking at the conglomerates in the landing site, which is known as Bradbury Landing, was really just key in making this fluvial interpretation. But conglomerates have actually been observed elsewhere in Gale crater since the landing site, but they have not been systematically characterized for their class rounding to kind of continue the story and the narrative of looking at fluvial transport in Gale crater. So that's what my work is ultimately aiming to do, is expand upon these analysis and compare the rounding of class elsewhere in Gale crater to see what kinds of spatial, temporal variation we might see in the rounding of clasts. So my work expands these analyses on rounding to two other locations. So Bradbury Landing is part of Gale crater known as Bradbury Rise. It's an elevated just region of topography, and the rover traversed along it. So you see the rover traverse right here in white, and Bradbury Landing is part of Bradbury Rise, but these are the conglomerate outcrops that I have been characterizing in addition to what was at the landing site. Another part of Gale crater is the Mount Sharp, which is in the middle of Gale crater, and it's a big mound of sediments that the rover is currently traversing up, and that is the big mountain that's in the middle. And what we see at the base of Mount Sharp were a few different areas known as the heterolithic units, where conglomerates are also were also imaged by the rover. These heterolithic units are really interesting because they represent out-of-place float rocks. So whereas in Bradbury Rise, there are stratigraphic context for the outcrops observed there, these are out-of-place blocks. So they're not actually occurring where they were originally deposited. So they lack the stratigraphic context to see relationships between other rock units to indicate the exact transport and depositional histories. But they still provide another snapshot at conglomerates that were likely transported somewhere upslope on Mount Sharp. So these are just blocks that probably were transported by some process that we don't quite know, but they provide another snapshot for conditions that were able to transport clasts of these sizes. So in order to answer these two questions that I have up here, I'm analyzing rounding. So the two questions are how rounding changes with distance from the Peace Vallis fan and what that can tell us about transport history. Since clasts that have been transported longer distances, you might expect to have more opportunities to have their corners abraded off. We might expect to see differences in rounding values for something that's been transported a shorter versus a longer distance. And we also want to characterize how rounding in Bradbury Rise compares to rounding in the heterolithic units, since they represent two different parts of Gale crater. So in order to actually characterize rounding, I have two different methods of doing it. There is the qualitative method, which is in red, and the quantitative method, which is in yellow. So I'll start with the quantitative method, which is a modified Wentworth roundness index. These are the same techniques that were used in that 2013 paper at the landing site. And what it does is it compares the ratio of, or it's a ratio between the diameter of the most angular corner of a clast to the diameter of the largest circle that you can fit inside a clast. So it's a lot of drawing circles in order to get this metric of how round a clast actually is. So you can see that the sharpest corner is right here, and this is the largest circle. So this gives you a ratio between 0 and 1, with 0 representing the most angular class and 1 representing a perfect circle, basically. We can also compare this to this qualitative method of doing rounding, because this, as you can tell, it might be kind of hard to pick out what the sharpest corner actually is. So this gives us an additional metric to compare it to, the Powers Roundness Index, which is from a 1953 paper. It's a well-used sedimentological technique of determining rounding, and you would visually assign a class a value between 1 and 6, with 1 being an angular class and 6 being a well-rounded class, and you can just visually assign it based on comparing it to this visual comparison chart, which is frequently done in the literature. And this is what some of the rocks actually look like, some of the conglomerates that are in Gale crater, and some of the median rounding values that were observed for them. So I placed these 4 example images to show an example of the variety of different rounding values that you can see for conglomerates in Gale. So this is an example of some low rounding values. The quantitative, so circle drawing method, yielded a value of 0.28, and then the quantitative method of assigning values got 3, versus all the way on this side, which is one of the more round median rounding value, the highest median rounding values that I've observed so far, which is actually one of the landing site outcrops that was in that 2013 paper at the landing site. So you can see that these are well-rounded class for this image. Like you can see that example of a well-rounded class right there, and you see a variety of different values. So this is what that data actually looks like plotted against elevation. This preliminary result is that most rocks have kind of this range between 0.4 and 0.5 for their median rounding values, or 3 to 5 when they're assigned qualitatively. So that's a subangular to rounded category. And I've plotted these values against elevation because elevation is helpful in a stratigraphic context. It gives us context for the temporal and spatial distribution of these conglomerates. So Bradbury Rise is actually an area of topographic high. So the further we've traveled away from the Peace Vallis fan is represented by higher elevations. So that's why we have elevation plotted on the y-axis there, and then the 2 different techniques here. So the second result I have here, still based off of the same plots, is that, oh, excuse me, let me go back here. I also wanted to point out that I included the heterolithic units here without actually assigning them an elevation value. So you can see they're omitted right here because they do not have that stratigraphic context. They're out of place float blocks. So I didn't want to assign a value, but we just generally have them on this chart to compare the values to the remainder of Bradbury Rise. So that's what's going on there. So another thing that we see is that when we compare the 2 techniques here of the quantitative and qualitative methods, there appears to be a more consistent trend when we're looking at the median Powers rounding values here. So you see a lot of 4s right here across different elevations, versus there's a bit more of a spread in the Wentworth rounding values right here. So this already gives us an insight into the comparison of these 2 techniques and the data that we're getting from the 2 of them. And what you can also see is that there are generally lower values for the heterolithic units. You can see them grouped here. There's actually 2 points represented right here. They just overlap since they're at the same elevation. But we ultimately see slightly lower values for the heterolithic units that distinguish them as a separate unit on this chart as well. So ultimately, the differences that we're seeing through the 2 different techniques might be a good reflection of the limitations of resolution. These images that I've been using to do rounding analysis have been based on 2 different cameras, Mastcam and Molly, that are both on the Curiosity rover, which provide images at different resolutions based on which camera we're actually using, and also how far away an outcrop was imaged from. So those are the 2 factors that can contribute to different resolution effects. So high resolution is reflected by the lower values down here. So these are the highest resolution images, versus lower resolutions, which are going to be from the Mastcam instrument, or higher up here. And you can see that despite having a range of different resolutions available for these points right here, I've consistently plotted them as having a median value of 4, which means that when we see the spread right here, this seems to be less sensitive to the effects of resolution in assigning values. Sometimes it's just easier to assign something when you can't necessarily detect what the actual pixel is, but you can see it, which appears to be one of the influences that are happening right here. To answer the 2 questions that I proposed at the beginning, these are some of the insights that these different results give us. So to think about how rounding changes with distance from the Peace Vallis fan and the transport history that might indicate, rounding is variable across different elevations. So there isn't a consistent trend necessarily of seeing, okay, at higher elevations, we definitively see rounder or less round class. Instead, what this might indicate is that there are multiple different source regions. So if the actual channels that we're transporting class to the Peace Vallis fan and the associated conglomerates. We're sampling from different parts of the catchment area. They might reflect different rounding values because they have been transported different distances. Or this could also indicate multiple transport mechanisms. Something that I didn't mention earlier is that conglomerates don't have to be fluvially transported. These larger clasts can be mobilized by other mechanisms such as debris flows, so gravity being the primary influence, water still playing a role, but these two different techniques can be distinguished between the roundness and the grain size distribution. So it's possible that there are multiple transport mechanisms happening here, referring to the different angularities of different clasts. And for the second question of how rounding compares between Bradbury Rise and the heterolithic units, we did see that more distinct group of lower values for the heterolithic units. So they appear to be more angular with a medium subangular value. So if they're fluvial, that would probably represent a shorter transport distance. They haven't had enough as much transport distance to round across. Or if they are not fluvial, it's possible that they are debris flows because we see those more angular clasts. So I'm looking forward to expanding upon these analyses, definitely characterizing more elevations to add to those plots because there might be more of a story to tell as we include more elevations on them and seeing what trends might come about. And I'm also getting really interested in the effects of composition on these rocks. Different rocks have different hardnesses, which can really influence how an actual clast rounds over time. So I'm hoping to supplement that with field work and comparing actually different lithologies and seeing how they round in the field and using that to interpret my results in Gale Crater as well. So thank you so much, and I look forward to your questions.
Any questions? Don't.
Hey, so those that relationship between rounding and resolution. So that resolution was in like pixels per millimeter on the sample or whatever. That's dependent on both the camera and the distance from the camera. Is there something systematic about like how likely you are to approach different things? Could there be something about like terrain that contains one type of thing versus terrain that contains the other that could potentially explain a slope in there too?
That's a good question. Thank you. So I think it's a good point. The reason why we have lower resolution values for a lot of these Bradbury Rise conglomerates in particular is because they were specifically imaged far away because we were really booking it across Bradbury Rise. And it was just opportunistic. Like we saw some conglomerate outcrops kind of just in the background and didn't necessarily come up to them. They weren't the primary objective to analyze them. We more opportunistically imaged them. So it's true. Maybe like if there was rougher terrain, it would make a certain conglomerate outcrop harder to encounter. But we haven't seen conglomerates particularly recently, but that would be something that I would advocate for is like get close to the conglomerates and see like just get as close as possible if it's safe for the rover, obviously. Does that answer your question? Thank you.
Hey, Pedro Montalvo from Auburn University. I'm a geologist and I love seeing rocks.
Yay.
So great talk. So my question is, are these conglomerates constrained, like the location constrained inside the crater? Or can you find them outside of the crater or anywhere else, like outside a crater, like in Mars, for instance?
Yeah, so that's a good question. The reason why these conglomerates were so exciting is because you kind of have to have a rover or some on the ground presence in order to image them. Since we're looking at clasts that are 2 millimeters in diameter around that range, that's not something that you would be able to hope to resolve from orbital imagery. Like HiRISE would give you up to 1 meter resolution, which is the highest resolution imagery we have of the Martian surface right now. So it would definitely take more rover work to detect where else they would be present on Mars. But we know that since they appear to be affiliated with this alluvial fan right here, and we do see fans elsewhere in Mars in other craters, and just like there's maps of the distributions of fans on Mars. Those would be places where we would want to go and send rovers to confirm the presence of conglomerates, but I don't think you would necessarily expect to be able to see exactly where they are unless you had that on the ground presence.
I have another question. If
Yes, go for it.
So have you thought about comparing like boulder size roundness versus
the size of the class versus roundness?
yeah,
Yes,
like
that is active work in our research group. Dr. Rivera Hernandez here is shaking her head because that's a big part of this project as well. And actually, Is she here? Haley was helping us with characterizing grain size roundness and grain size as well. So that's a data set that we have to compare to the rounding of class as well. So thank you, Haley, for supporting this work with that data.
Thank you. So I have one, maybe a bit of a dumb question, but are there other non-aqueous processes that could influence the shape and the size of these conglomerates?
I think like weathering just in situ after they've been deposited can definitely influence rounding. On Earth, if you see like secondary minerals being deposited, that can definitely influence the way that something weathers and how round it can be over time. So I think that that's something to consider that I have not yet done. It's kind of hard to constrain for class that are actually in place inside a conglomerate to see how like weathered they might be versus a pebble that's just sitting on its own in the ground. But that's something that comes to mind as like an additional process, or just aeolian abrasion over time as well, since they're old.
Really great talk, Tatiana. Thank you so much. As someone who spent a lifetime measuring grains myself,
Yes.
like I really respect what you've done here. So two questions. One is, so number one, I recognize this PhD and I tell my PhD students, like, you can't stay forever, so you can't take on all the questions, but have you thought about comparing to the conglomerates in Jezero that Perseverance has been imaging? So that'll be my first question.
Yes, definitely. So Jezero crater is where the Perseverance rover is, which has been on Mars since 2021, for those of you that are keeping track of your Mars missions. And there have been really big boulders that are rounded that have been observed kind of from a distance in Jezero. And I would love to just do like a quick comparison of the rounding values. I think 2021 paper already kind of did some preliminary work on characterizing the rounding, but I think having a consistent operator, compare them, these values can kind of range from person to person. I would like to just see how my values match those values and get a better sense of like the transport history of those boulders as well, since you need a lot of energy to mobilize a boulder versus a piece of gravel.
Yeah, and I think we're definitely seeing a range, right, of like gravelly conglomerates versus like, yeah, boulder sized clasts. And then second question, on one of your figures, you were showing error bars.
Yes.
Yeah. There it is. So I don't do like the statistics side of things, but would a statistician look at this and be like, there's no difference because of your error bars? And I'm just curious about how you're approaching this.
Yeah, so these error bars, I should have clarified, thank you for pointing them out, represent the total range of values that are characterized for one class or for one outcrop. So about 100 points is what I used to determine the median of the rounding values. So like if I look at this rock, I established 100 class that I would measure. So the median is the median of all of those, and the error bars represents the lowest and highest values, which I know might not be the way that information is typically conveyed. This is like stuff I just added here. But this can also help just give insight into like if you see only rounded class, like for this right here, where you only see a range between 3 and 5, that's indicative that everything's kind of been subjected to the same rounding processes, versus here they are a little wider. So it would take some additional analyses of maybe like some of the sedimentary fabrics that we see to get further like a fluvial interpretation. If you see like really angular class incorporated into some of these that might have higher actual median rounding values.
So how did you select the 100 class you were going to characterize in each image?
I overlay just a systematic grid over here, and then if a point intersects with a class, then that's something that I measure. Thanks. Thank you for your questions.
Yeah, these are great questions, everybody. So I think we're going to have our break now.
Yes.
So let's thank all of our speakers from the morning session. Thank you. There's still some breakfast left in the back. We'll have about 5 minutes here for break, or 5 minutes. We'll give it about 5 minutes, and then we'll be back for our bio session. Thanks.
Awesome. Yeah, so let's.
Where do I stick it?
Yeah, you can drag it.
Give session 2.
Oh, yeah. That's pretty sweet.
Yeah, I've been throwing together a grant all week, and so I literally put this together last night. Yeah. Well, Good thing is, I was able to take figures from the grants and put it right into here. Some of the maps are straight off of the computer yesterday.
Okay, so basically, you'll just use this clicker.
Yeah, yeah.
I'll have this ready to go, and you can just use that button right there as the laser
Yeah, yeah.
pointer as well.
Do good. Oh, not many people go backwards.
Yeah, I know, exactly. So I'll probably be right here and I can cue you, when it's
yeah.
the 10-minute mark, with 2
Yeah.
minutes to go.
Yeah, my presentations are weird. I like, instead of anything like animations, I have like lots of barely different slides from each other.
Okay,
So when it's
Yeah,
so
no,
when you
that'll
suddenly
work.
see, oh my God, 35 slides. That's
yeah.
not actually.
Hello. Can you hear me?
Oh, great, never mind.
Screen wasn't working for a while.
Did you just move the mouse? Okay, cool. Well, it was good. Something happened to make this toolbar go away, and we're like, great.
I think it's just.
Which I think we should turn the transcription.
Oh, the mouse is over here.
Oh, okay, yeah.
Yeah.
I would stutter.
I think.
Am I just doing what Mike did last?
Okay, excuse me, everyone. Hello. Can I get everyone's attention, please? We will be beginning our next session, so if everyone can please be reseated, and we'll get started. Okay, so our next session will be about proteins. We have two speakers presenting today. Our first speaker up is Dr. Claudia Alvarez Gareño. So with that, I'll let Claudia take over and go ahead.
Yes, thank you. Oh, I think I shared wrong. 1 second.
1 second, Claudia. I think we need to get your audio set up here.
Good. Let me know if you hear me.
Can you say a word for us, Claudia?
Yes. Can you hear me? Hello?
Okay.
Can you hear me?
There we go. All right. Can you say another word for us, Claudia?
Yes, sure. Can you hear me?
Awesome. Yes. Okay. You are good to go. Thank you.
Okay, so today I want to talk about the evolution of protein structure. And the idea behind this research is that nature has been able to evolve all these proteins that form macromolecular complexes that are very impressive. So here we have some of the examples of all these macromolecular complexes that we observe today. And the question is, how did this happen? In the beginning, what was happening to allow the evolution of these structures? And I want to give an example of or illustrate what we want to do with these proteins. So when we have a historical record, we can learn something about the age of this historical record, but we can also learn of things that were happening before this record existed. So we may have a date for the invention of something, but then, for example, in this clay tablet, we can assume that by the time this clay tablet was invented, the technology to make it possible already existed. So that's the kind of things we want to do, but not with historical records like this, but with proteins. So instead of using fossils or any type of existing structure, what we use today is a comparison of macromolecules that exist in living organisms and try to get all these molecules that we can trace back to the last universal common ancestor, or as we call it, LUCA. And the last universal common ancestor is this population of organisms from which all life that we know today emerged. So once we have these molecules that we know have been inherited from generation to generation for the past 3.8 billion years, what we want to do is understand what was happening before their emergence. So we want to go beyond the last universal common ancestor. And to go beyond the last universal common ancestor, one of the tools we can use is understand duplication events. Because if we have a duplicated gene or protein, we can infer that there was an ancestor of this duplicated event that existed before the duplication. So paralogs or duplications are one of the best tools to start looking into processes that happened before the divergence between archaea and bacteria. So there are many macromolecules that we can study. The one that I am studying are protein domains, and protein domains are formed by different structural units called protein domains. And there are only about 1,000 or 2,000 protein domains that are known today. So over 4 billion years of evolution, we only have the record of a few structures that have persisted. So how can nature get to these structures? And it's a very difficult question to ask, because most combinations of amino acids will not get a stable protein structure. So most combinations of the 20 amino acids will generate conformations that are not stable. So one of the hypotheses that was used before was that all the protein domains that exist today had an independent origin. So there were around 1,000 or 2,000 independent origins, and that nature came with the combination of amino acids to form these stable structures many times. On the other hand, there are also hypotheses that say that all the protein structures that we know today are related to each other. But really, the way we understood evolution of proteins was more on the side of the many times being invented. But one of the things that started happening when we were able to compare proteins against each other was that we realized that many of these sequences of the proteins that exist today share sequence similarity and structure similarity, but not overall, not in the entire protein, just in local regions. And that made us question, what does it mean? Does it mean that only these really small regions share ancestry and everything else evolved very rapidly at the origins of times of life? So what we did is we took a combination of these things. We took a combination of the repertoire of proteins that we know are present in the last universal common ancestor, and also these comparisons between many protein sequences and structures, and we tried to make sense of the information we were getting. So one of the ways we can understand how two proteins with different structures are related is taking a very simple example. And the most simple example of sequences that are related but that have a different structure is something that we call circular permutation. So in a circular permutation, we will have a very similar overall three-dimensional structure, but the individual elements in these structures seem to be rearranged. So in the structure of the left, we have the blue elements on the beginning of the protein, and on the structure on the right, we have them on the right hand of the structure. So we could think, how can we get from the structure on the left to the structure on the right? And one of the options is maybe you can just cut this region in blue and paste it at the end of the sequence. But that's not really how gene evolution happens. So when these proteins evolved, there is no copy pasting of small structural elements and jumping around the genome of the organisms. What really happens that allows the evolution of these circular permutations is that when you have a gene duplication, a tandem gene duplication, you can then have an emerging property that will make a new fold emerge from this new combination of structural elements, and then the termini of this protein are lost. And in this way, we can get a rearrangement of the structural elements without actually cutting and pasting anything. So we do understand how this modification of the structure can happen in simple examples, but can we see that in a more complex example? So is it possible to see relationships between folds that are more diverging than circularly permutant proteins? So this is one of the protocols that I developed using many bioinformatics tools that were already available. And the idea is to go from the sequence information to the structure information and then iterate in the process to understand very early evolutionary events. Now, the next thing that we did is we took some of the proteins that we know were already present at the last universal common ancestor, and maybe even long before archaea and bacteria diverged. So one of the structures that we see everywhere in proteins of the translation system is the small beta barrel domain. And here in orange, I am showing you where we find it within the ribosome or within other proteins. And these are some of the most conserved macromolecular complexes that we have today. We know they were present already at the last universal common ancestor. And what we did is a all versus all comparison of the sequences of these small beta barrels. There are three categories of small beta barrels that I'm showing you here. One is called SH3, the other is OB, and the other is CLB. And the interesting results are all the blue squares where we see similarities crossing these categories. So the similarities that go between an SH3 and an OB and an SH3 and a cradle loop barrel, which is CLB. So what we see is that there are many of these cross-category similarities, and this is comparing the sequences. So what does it mean? So the SH3 is this structure on the left, and the OB is the one on the right, and they are very, very similar. Here it will almost look like a circular permutation, but in a circular permutation will typically require only one cut and paste event for it to be called circular permutation. Here we need two, so it's increasing the complexity by one. And we do see sequence similarities between them. Then we added a new structure, which is the cradle loop barrel here. And in this cradle loop barrel, it's not only a rearrangement of the elements, we have new elements coming. So once we had the structure comparisons and the sequence comparisons ready, we started doing an analysis to see if we can tell the story of these relationships. So what we found is that it's not too complicated. You can take the copy of two SH3 domains, so two SH3 domains, and form a new fold, which will be what we call today OB. So the names are not very informative here. These are only two types of barrels. But the idea here is that the duplication of one ancestral gene can generate a completely new structure. And then in the case of the cradle loop barrel, which has an increase in the number of secondary structural elements, we can take a copy of the OB and generate a new structure. And this all happens without having to ask for evolution to do something with foresight. So it just happens by a duplication and evolution and divergence. The other thing that we did is we use one technological advancement that has changed completely the field, which is all these artificial intelligence algorithms that allow us to predict the structure of a protein. And we took AlphaFold to predict if our duplication events could really generate a new fold. And by doing the experiment, we got that a duplication of SH3 generates an OV, and the duplication of an OV generates a cradle loop barrel. So this is the diagram of how we think it happened. And if I just summarize what I have said, we have a new mechanism that can explain how two different faults are related, and it acts at the level of the domain. When we combine domains, we will not have always a repetition of the domain. Sometimes we will have an emerging new protein domain, and this will allow us to go even further in time before the emergence of the first proteins. So that's what I have for you today. Thank you.
Thank you so much for that. It was a wonderful talk. We have time for at least one question, if anyone has one. Yes.
Hello. Love it. I saw that you had a I saw that you have a apparent hypothesized trajectory about which of those domains could have come from which ones, and I was wondering if there is a if there's any tendency in terms of where those proteins are or what they are involved in that might tell you if something is older, if some process is older than something else.
Yes, a great question. So I didn't show it here, but we have an idea of what elements in RNA are older in the ribosome. So for that, we have a very good timeline figured out. And if we take these folds and locate them in the ribosome, we see that it is as our hypothesis goes, the SH3 is in the oldest part of the ribosome and the OV is in the newest part of the ribosome. So at least for these, there is correlation between our hypothesis and what we see in very ancient structures.
Okay, amazing. We also have an online question. Saritha Dante asked, will linear structure also appear?
Let me read. I didn't get the question.
The question is, will linear structure also appear?
Yeah. That's yes. Well, yeah. Yeah, we didn't study that. We only focused on the 3D correlation, but our hypothesis also would explain the linear structure. We didn't study intrinsically disordered proteins. Is that what this is about? I'm sorry, I don't know how to stop this.
We can. It's yeah. Go on. Okay, yes, we need to move on to the next speaker.
Yeah.
Thank you again for answering the questions in your presentation.
Thank you.
So next up, we have Dr. Anthony Bernetti, who will be also talking about proteins, and I'll let him take it away.
All right, is this on? Yes, it is. All right, so I will be talking about a very specific set of proteins today. I'll be talking about oxygen binding proteins, how they relate to facilitated diffusion, and the evolution of multicellularity. So all of this is really motivated by one big question. What is the role of Earth's oxygenation in the evolution of multicellularity? So many of you are probably aware, Earth has basically been three different worlds with three different states in terms of how its atmosphere has worked. You've had the basically zero oxygen, Hadean and Archean. You've had a low but present oxygen in the Proterozoic, and in the last 600-ish million years, you've had the Phanerozoic. And the Phanerozoic, in a couple of ways, seems kind of special. I mean, they call the Proterozoic the boring billion sometimes. So importantly, this is not going. I'll use the arrow keys. Nothing is happening. There we go. Can I move it? Yes, I can. Okay. So importantly, in the Phanerozoic, you've had an extreme diversity of multicellular life and extreme amounts of multicellular biomass of all kinds of diversity. Multicellularity has evolved many times. 6 or so times people tend to think it's particularly interesting and complicated, but dozens of times that it's evolved at all. And it may not be a coincidence that a lot of this has occurred in the Phanerozoic when the oxygen is high. And so our lab actually, we do evolution experiments with something called snowflake yeast. We've done previous experiments supporting the role of increased oxygen in multicellular evolution. This is a nice, simple model system for examining simple multicellularity. These are just yeast cells that have a single mutation that prevents the daughter cells from completely separating from their mother. And so you get this nice little nugget of yeast, and you can just do laboratory evolution for increased size via settling selection. You just grow them, put them out on the bench, let them sink, and use the ones that sink the fastest to found the next generation. And this is some previous work from Ozon in our lab, amazing scientist. He's done some evolution experiments where you take these yeast, you force them to grow aerobically. If you do that in low oxygen, they stay small. If you supplement the oxygen, they get big. So we have reason to think that we've done previous work on this, suggesting that oxygen is important. But interestingly, you look at the evolution of animals, and at the same time oxygen rose, oxygen carrier proteins evolved. Everybody knows hemoglobin. That's one of a diversity of globins. You've got the hemoglobin that circulates in blood, myoglobin that's stationary in tissue, but there's others. You've got your hemorethrins, 2 different hemocyanins. They're really diverse. But interesting question is, why did these evolve when oxygen was already high? Because you can point to where they occur in animal evolution, and it's really after the oxygenation has occurred. So to look at this, we're going to focus on myoglobins, not hemoglobins. Everybody loves hemoglobin, but that circulates in blood. But early globins were stationary, sitting around in tissue. Circulation is a late invention, and it was invented multiple times, even multiple times within the vertebrates. You go across all the animals, it's even more. This is a nice little tree of different globin genes in vertebrates, and you can see the inventions of circulation in red. So, and it's not just the globins. Other oxygen binding molecules evolved independently around this time too, circa the origin of the bilateria. And so what are these stationary oxygen binding proteins doing? Turns out it's facilitated diffusion. You've got the exterior of the organism with lots of oxygen. The proteins are able to take it up in the exterior, just diffuse without bulk flow, and give it up where oxygen is low. And it's basically you just add diffusible oxygen binding sites in addition to the diffusible oxygen. And this allows increase of oxygen flux even when the oxygen binding proteins aren't flowing. So my goal is to use snowflake yeast to explore the role of these oxygen binding proteins in the evolution of multicellularity. So we're going to express these relevant proteins in the snowflake yeast, expose them to relevant oxygen levels in clusters of different sizes, look at the physiological effects, and perform some laboratory evolution experiments. And we've done so. So we've expressed myoglobin. This is a nice monomeric single globin domain, holds a heme group with an iron. It can transport 1 oxygen molecule. And we use the one from sperm whales. Just because due to accidents of history back in the pre-genomic days, you characterize the proteins from sources that were really dense. And it turns out whale meat has way more myoglobin than beef. And turns out we express it, the yeast get pink. So seems to be working. We also expressed myohemerythrin. This is less well studied. It's also possibly present right at the base of the animals. People disagree. They don't have the sequence determinants of binding down as good. It's also a nice monomer, easy to work with, and it directly holds iron atoms instead of using heme. And we use it from a sequence from peanut worm. It's just the best characterized one. And I wish I had a nice picture of this one, but it turns out that this one, when it's deoxygenated, is colorless, and that's what happens when you spin down a pellet to take a picture, so it looks boring. But so we've done some experiments, and I just have to plug Whitney Wong, the technician who was working with this on me. If any experiment here worked, it's because she made it work. So she's gone on to other things, but her work was fantastic. So we've done some physiological analysis of the O2 binding proteins. So we expressed these proteins in normal snowflakes and some small snowflakes in an intermediate oxygen level and a supplemental oxygen level. And we competed these yeast with yeast that didn't express these proteins. And it turns out that for the small snowflakes, with supplemental oxygen, there's really no big significant effect. But at the lower intermediate oxygen levels, they have a bit of a selective advantage when they express these globins. Then you take the larger yeast, they have some selective advantage at the intermediate oxygen levels, but you go to the supplemental oxygen and the advantage more than doubles. So this is interesting. These proteins are having a larger effect in higher oxygen. And so we also then performed some laboratory evolution experiments. And for this, again, we just daily passage these yeast in oxygen limited environments. The oxygen limitation imposes selection to use the oxygen well. And then we also select them for rapid sinking. And that settling selection produces a selective pressure for increased size. And so we have a 6-month-long laboratory evolution experiment. As laboratory evolution experiments go, we've done much longer in other circumstances. Ozone has done an amazing one that's more than 4 years old now. But even just after 6 months, it turned out all the populations we looked at, they bifurcated. They turned into a coexisting large and small cluster population. And this is somewhat expected. This sort of thing happens when you evolve yeast under low oxygen environments. But we then took a large isolate and a small isolate from each population. And it turned out that we then measured the expression of these oxygen binding proteins. And it turns out the large isolates nearly universally significantly increased their expression of the oxygen binding protein compared to the small isolates, which for the most part were like their ancestor, except for this one weirdo. For some reason, one small isolate decided to really turn up the expression. So apparent, and also I will note the myoglobin lines, they got visibly pink. The evolving populations were colorful. So we can conclude from this that facilitated oxygen diffusion is beneficial for the evolution of increased multicellular size. It's more helpful when oxygen is high than when it's low, and it's more helpful for large organisms than for small organisms. And so together, this suggests that the oxygen binding proteins best represent a way to circumvent anatomical limits to oxygen diffusion once oxygen is already plentiful, which would fit with when it appeared and the organisms in which it appeared. So, and continuing on, some very fresh data here. These models are literally still running on the lab computer. We're looking to model facilitated diffusion computationally, because snowflake yeast, they have specific biological properties, and not all organisms will share this. So, and over the course of their growth cycles, it's hard to pin down particular oxygen levels. Things are constantly changing. So we want to broaden applicability and model this at different parameters that might be relevant. And so we just do this nice numerical solution differential equations in Julia, modeling diffusion of the carriers, diffusion of the oxygen, the consumption. And we can also modify the geometry. We're focusing on spheres, cylinders, and planes. Basically, there's sort of broad geometry types. You might have something floating around in the water, filamentous algae, or say a flatworm. And I got to thank Pablo Bravo for some coding on this. He is the reason that this code actually works. So some preliminary results. We can these are from modeling spherically symmetrical organisms with yeast-like parameters. You got some large and small clusters at different oxygen levels. The globins increase the penetration of oxygen to a deeper depth. Makes sense. And then we can sort of integrate over this and get the total metabolism of the organism, and we see some fun results. Here's some results of 50 micron and 5 micron models at different oxygen levels for different quantities of globin. And in short, large organisms always have more benefit than small organisms from the globin. And large organisms benefit more at higher oxygen levels than very low, while the small ones benefit more at low than high. It's consistent with what we're seeing. The numbers are a little weird. We're still zeroing in on the parameters for the model, but we think we're seeing something appropriate. And we're also exploring the effect of different geometries. This is a heat map pulled off of the computer yesterday for spherical organisms and planar organisms at different oxygen levels and different radii. And the interesting thing, apparently, planar organisms act like larger spherical organisms when it comes to the effect of the globins. So this might have something to do with which organisms this might be likely to evolve in. We're still working on it. Lots of parameters to explore. So continuing on, again, we're exploring the parameter space. We want to try to get parameters appropriate from like animals to algae. We're going to express additional oxygen binding proteins. Some people have done ancestral reconstruction of 500 million year old globins that were in animals. We want to express these. And we want to extend these laboratory evolution experiments because in our experience, interesting evolution can happen 2 years into one of these things. So we're excited to see where this goes. And also, I want to explore a second molecular mechanism for circumventing this oxygen limitation, and that is phototrophy. It's striking, all of these multicells in sort of the Proterozoic, they're that appeared before the big rise in oxygen, they're all algae, they're all phototrophs. I don't think this is a coincidence. I think this is another way to get energy into those deep tissues. And I'm working with Autumn Peterson in our lab. We've introduced microbial rhodopsins, a nice, simple phototrophic system into the yeast. We're able to show that they're injecting energy into their metabolism. We're really excited. So I'd like to thank everyone who's ever funded me, the people, the people who have been doing the work with me, and everyone in the lab, and I'll take any questions.
Okay, awesome. Looks like we have one question. My way over.
Hi, thank you for that really nice talk. I'm really curious about that weirdo. What?
Yeah.
What do you think? Why do you think it's there?
We haven't looked at it closely. What I suspect that something that we see sometimes in these populations, we're growing diploid yeast. Sometimes they sporulate and turn haploid. And because of some fine details of yeast biology, the haploid clusters are way smaller than the diploid clusters. We're wondering if that's actually a large one that had a high expression that suddenly turned haploid and got really small and is like changing its ecology. We're not sure. That would be my hypothesis, but we need to check.
Any other questions? Okay.
Hi, they're great talk. So I'm Philip Stansel, physics and astronomy UGA. So I'm going to ask some dumb questions.
Okay.
And know nothing about biology. Would you include like the solar irradiation in these simulations?
So the models so far are just of oxygen diffusion.
Okay.
I want to model adding a phototrophic metabolism once we've done some experiments and seen if we can provide an advantage to multicellularity from our artificial induction of phototrophy. I would be very interested in adding a model of light penetration and light intensity to see if we can get some phototrophic metabolism also doing this. I'm interested in that too.
Okay, and following on that, the period of the Earth evolved, over this 4.5 million years.
Yeah.
At the Great Oxidation event, it was about 16-hour days.
Mhm.
So I'm wondering if there may be some variation on, light period.
Yeah,
Daylight period.
the circumstances under which phototrophy is useful or not.
Yeah.
I've read a paper a while back where someone showed that faster days, meaning you need to adapt to different light intensities faster, means that phototrophy is less useful. So
see.
so that could be interesting. Yeah.
Okay. Sure.
Great talk, Tony. Have you, like your big evolved clusters of yeast, can you like kill them with carbon monoxide?
It's very, the thing about yeast is they don't need to respire. Yeast, if you give yeast glucose, they will actually turn off their mitochondria because they actually prefer to ferment. In these circumstances, we've actually been growing them in glycerol where they're forced to breathe. And so, yeah, I suppose that I suppose we could kill them with carbon monoxide, but I think that would function by killing the terminal oxidase as well. So I'm not sure how much. On the one hand, we could destroy the oxygen diffusion. On the other hand, I think destroying the oxygen diffusion might also kill the terminal oxidase. So.
Okay. It seems that we have a question from Claudia online. So Claudia, if you want to unmute yourself and ask your question, go ahead.
Yeah, thank you. So great talk. And I actually worked on hemerythrin during my PhD, and I remember that one of the things that can happen is that when oxygen is released, instead of oxygen, you will have hydrogen peroxide. So with these oxygen binding proteins, there's also now a risk to having reactive oxygen species. Do you also take that into account in your models of evolution?
I do not. That's interesting. I don't know if that would be. I should look into that. If these oxygen binding proteins also bind the interesting reactive species. Yeah, that could be interesting.
Thank you, everyone. That concludes session 2 for today. So lunch is being set up outside this room, and you can all follow out that door in order to get lunch. And we'll be on lunch until 12:30, I believe. Yes.
Yes, and at 12:30, we will have our first plenary talk with Dr. Amy Williams. So please catch at 12:30 Eastern Time. We will be back online for the folks that are online as well. Thank you.